right? And you guys are going to ask some questions, and to the best of my ability, I will answer them. And you're at Landis Homes in Pennsylvania, a retirement center, I guess, a retirement village of some kind. And what is it called?
A community.
A community. That's
a continuing care retirement community.
Excellent.
Pollyann and Ken are still stuck, it looks
They
like.
are. So if they can hear us, they can go and come back again, and they probably will do that. So yeah, so Ken Brown and I have known each other for a number of years. His son, two of his sons, I believe, went to my school at Gordon College where I teach. And I've gotten to know him through doing various projects. Good to have you back, Ken and Pollyann. And anyway, so we're here to answer your questions. As a biologist, I've studied various viruses, including in my postdoc, cytomegalovirus, and as a teacher of undergrads teaching general biology and biochemistry and immunology, I have a good storehouse of information, background information that can be of use in this current pandemic situation. So yes, today's date is May 7th, and we are, I believe, at least 2 months in this situation here in the U.S. It was early March when I was on a spring break trip to Florida, and this was starting to be known, and I'd taken some precautions and brought some ethanol and things with us on the trip, and we were on the way back at the end of spring break. We knew this was serious, and we were starting to be more concerned. But time has gone by, and I see you guys. We talked. What was the previous date of our previous discussion?
Don't know.
If I had to guess, I'd say it was the end of March, probably. So it's been almost a couple of months, well, a month and a half, and you have more questions. So I'm here for you.
Well, I'll give you a peripheral one that Joe and I talked about earlier, because hopefully it'll be relevant. We don't have any RNA vaccines for humans. Why would it be good to have them, and why would it be good not to? I realize that's more technical than some people would like, but it's important because there are DNA vaccines, there are vectored vaccines where chunks of DNA get taken into the central nervous system and so on. So just give us a quick rundown on that issue.
Yeah, and I think Dave had previously, before we started recording, he had asked me about that question. Maybe it was Joe. About RNA viruses versus DNA viruses, and then connecting that with different kinds of vaccines. I think maybe there's a little confusion about the vaccine question. Let's save the virus discussion, and we'll just focus on the vaccine question, because vaccines come in a variety of types. The goal of a vaccine is to train your immune system in a very specific way so that you develop antibodies and T cells that are highly specific down to the molecular level to the particular bug. We'll just call it a bug, whether it be a bacteria or virus, particular pathogen that you're fighting off. And so our immune system's made up of a bunch of different kinds of cells, as you know, and the cells that are responsible for this very specific learned kind of immunity with which you inherit, you keep the memory alive of that vaccine for years to come, typically. That is accomplished by B and T cells, these particular kinds of cells. B cells making antibodies, T cells fighting off these virus and viral infected cells all on their own. They have very specific receptors. So again, back to the vaccine. So your vaccine needs to train these B and T cells very precisely and specifically. So to do this, you need to deliver antigen to your body in such a way that it causes an immune response to happen. And the antigen can be in any form, but it needs to be the three-dimensional shape of whatever it is that the bacteria or the virus that you're trying to fight off It has to have that same three-dimensional shape, that same molecule basically has to be in the vaccine. So lots of vaccines are just components of viruses. Like in this case, it would be parts of the coat, parts of the spike protein that protrudes from the coronavirus. They actually cut and paste the DNA encoding that. They would use DNA because ultimately that DNA gets made into protein. It has to go through the intermediate of RNA to be made into protein. That's beside the point. You take the gene, basically encoding that surface protein, and you manufacture some protein artificially and inject that into a person, and that could be a vaccine. Because it contains the component of the virus that you really want to make antibodies against, which is the outer surface knob spike protein. To make the vaccine effective, though, you can't just deliver the protein. You have to usually add some other components that make the immune system see this as like a dangerous substance and actually get the cells activated. So you can use various tricks to do that as well. So this is the process of vaccine development, is to figure out what is the antigen, the protein, or the molecule that we want to be in the body that's foreign and that your immune system will respond to. What is that going to be? How do we choose it correctly? And how do we get the body to fight it off effectively and not just kind of see this as nothing, no big deal, and not respond? For example, we eat food which contains foreign proteins, but we don't make antibodies against it typically. So you could, if you injected someone with some rice protein with the right kind of nasty molecules, say from bacteria, you would all of a sudden become allergic to rice or immune to, you'd have a vaccine against rice, against your food, and it would be a bad thing. So the goal of the vaccine development is to get some foreign protein in your body in such a form that your immune system responds. So that's what many, many groups are doing right now. And in terms of an RNA vaccine, that doesn't really make sense because, again, you just need to make this protein in such a way that it's delivered into the body. You don't usually ever make a vaccine against a nucleic acid like a DNA or RNA. That wouldn't really make sense. It's against some component of a bacteria or a virus, and 99% of the time it's a protein component. I'll stop there and ask if there are any clarifications needed.
Yeah, just an ancillary question was, what makes it such a long process? Is it trying to figure out what you need to send in, or is it making the thing, or what makes the process so long? Why are they talking about, usually 1.5 to 2 years to get a vaccine?
I have a lot of good info on that from what I've heard. In the past, it was really hard to just make that protein and even to figure out in animals which protein to use. But now we have a lot more tools that speed up that end of the process. So it's pretty easy to select some things that are likely to be immunogenic, unusual, and you can search the databases and make sure that there are not already similar sequences in human genes. You wouldn't want to inject a piece of the virus that resembles a normal human protein. That would be really, really bad, right? You would develop autoimmunity if you used that. So you can do that all with computers very quickly. And so that's the easy part. The thing that takes the most time is safety and efficacy studies in humans. Typically, they would do many months of animal studies and really get a good degree of confidence. They'd optimize the vaccine in animals. And then they would go to non-human primates, macaques or something, and then they would do human studies. They're already doing the human studies with this. They're pushing it at a really fast pace. And a group in Oxford is, I think, in the lead. And so they've already vaccinated a bunch of people with this new vaccine and another bunch of people with, I think, measles vaccine or a control vaccine, a different vaccine, right? And then they're allowing the people to just wander about, go shopping, and when enough of the people that have been vaccinated, presumably if it works, they would be in the control group, get coronavirus. They can go to the statistician and they can do a calculation to determine if it's statistically different between the two groups. You need to have a certain number of people get sick and right, in order to have a statistical, say there's thousands of people vaccinated and there's 40 people that all got sick in the the control group and none in the other group. Well, just from a gut, I'm saying that would sound pretty significant, right? Now, that takes a long time, especially if the curve is being really flattened and there's not a lot of infections going on in the population. So ironically, the more the curve is flattened, the longer in that model it would take to get the answer about whether the vaccine is effective. I will say one more further thing. Does that all make sense, what I just said?
Yeah.
Yeah. There's one other thing though. This was actually on Friday, May 1st on a show you might want to listen to. I think it's an NPR station that plays it in our area. It's a show called The World with Marco Werman. And they interviewed some people about the vaccine and the rate of getting a vaccine result. There's another way to test the vaccine. And Ken, you might be familiar with this. How could you test the effectiveness of a vaccine with, say, only 100 people? And you could tell within maybe a month's time whether it works or not.
Well, for one thing, you do an intentional infection study.
That's it. They call it a challenge trial. And the only problem is this is a pretty dangerous virus and a significant portion of people die from it. So then it becomes a really heavy duty ethical discussion. How do you guarantee with pretty high confidence that no one's actually going to die? You would choose some groups of people that you know are least likely to die. And you would, of course, they would have to be fully informed about the situation. But boy, that's a real bummer to be in the control group of that experiment, because basically you're going to get the coronavirus, you know.
Can I give you a historical perspective in one sentence?
Well, sure, and I'll just say that's currently done with flu viruses. They do that, challenge trials today. Go ahead, Ken.
Smallpox vaccine was done with a cowpox, which is a virus that looks like the smallpox vaccine. And they used a challenge study of one. It's an 8-year-old boy.
Phipps was named Phipps, I think, right?
I don't think that one was, but anyway, it's an 8-year-old boy, and that was several hundred years ago when you didn't have an IRB, an institutional review board.
And they used to use, of course, use prisoners for this. And then if people used to go to prison for things like financial debt and whatnot, and they would let them go if they, participated in the study. It was incentivizing them to. But anyway, so yeah, challenge studies could speed it up, but it's still being discussed and debated now. What would make a challenge study much more likely, and which would be if there were a therapy that was able to minimize the chance of death of someone in the study. So maybe the remdesivir would be an example. If that actually is working, then would have a greater chance of wanting to do this kind of study.
Let me see if I missed something here. If you intentionally start a person on a coronavirus, is that really different than sending them out into the population and hope that a good number do catch it?
The only difference is that it's much faster and in a much more controlled fashion. So you get your result a lot faster. Instead of just randomly waiting, retrospectively looking at the population, you already know that they've been challenged.
And it's an absolutely different ethical issue, because if you just let them live and they may or may not get exposed in the general population, that's a lot different than saying, okay, guys, 6 of you are going to get this and 6 of you are not going to get it.
Because you haven't done anything to increase their chance of getting infected by letting them wander wild and free.
Okay.
Yeah.
Well, morally, they're very different.
Yes. Yeah. Absolutely. So it'll be interesting to see what happens in time. Now, the other interesting thing, if you listen to that show, which I recommended to you, The World, apparently someone set up a website and there are thousands of people that are willing to be in that study. no shortage of people willing to, because if you think about it, even if a couple of people die in the study, if you move the vaccine forward by months, you could be saving tens of thousands or hundreds of thousands of other people's lives. So it's really hard to know where the ethics lie in that situation.
Mhm.
Certainly, it's like going up to the front line of a war, and you're likely to get shot, right? It's a very similar kind of a situation.
Craig, can you just walk me through that process again? Are you saying that there would be an injection of a live virus into?
Nope. No, they would probably just spritz it in their face. I don't know. I'm guessing they would probably instill it somehow in their lung or something. I don't know how they would deliver it, because that's one of the issues why that would make it very challenging, because it seems like some of the healthy young folk that died from it are ones that got a high dose. So you certainly wouldn't want to give them a high dose, but they should be able to titer it down so that they give them a very low dose, just enough for it to take hold in enough time for the immune system to catch in those control individuals who don't have protection. The other thing is you would certainly need to isolate them from everyone for a period of during their whole time that they're undergoing the study, because you couldn't give them the vaccine and then send them home to the family. So that's another complicating factor.
You would, what I would do is I would do it in a chamber with a vaporized virus at a certain pressure and certain volume, and then everybody gets exposed to the same dose. And you would do counts on the viral concentration at the end.
Yeah, you could do that, and then you have to decontaminate everything, though. You could probably also just like take a little drop of it and put it in their nose or something. It seems so catchy. If you can catch it from a fingerprint, from a hand transfer, wow, it's amazing. One of the more remarkable videos I saw recently was one of where someone used laser photography and were able to just show all the drops that come out of your mouth when you speak. And it occurs to me that if any of us has a computer laptop and you dim the light all the way down on the screen and you look at that thing, you'll notice that it's just literally covered with teeny tiny little spittle drops. So we do release droplets as we speak all the time, and these are the droplets that can carry these virus particles. And they're very tiny viruses, are really small. So great.
What is the minimum quantity of virus that will result in infection? One viral particle or one molecule, if I can use a chemical term, of virus, or
Yes, it
of
depends.
Avogadro's number, or
It depends on the virus. That's a great question. It totally depends on the virus. The number of viruses you need to catch hold in a person. It's not always just one. It can be that you need a certain number. But I believe one of the most contagious viruses there is the measles virus, and I think in that case one is enough.
Wow.
Because really it just hits one cell, gets in there, replicates, and then you have thousands and then thousands, and it's like it really ramps up quick. And the strategy of a virus is to multiply itself, to grow quick enough that it can propagate before you get really sick and are in bed. And so then it can spread itself. So that's the selection from the perspective of the virus. It's advantageous to quickly multiply and get out there in the world before you're bedridden or dead. And an ineffective and a less effective virus would be like the Ebola virus, where it actually doesn't spread very well because people are so sick. By the time they're contagious, they're already in bed, and it doesn't actually spread quite as well as this one.
When you use a word like "it wants to," what are you really saying?
No, it's a way of personifying something that has no personality or intention. It's a shortcut. It's not really obviously planning and plotting, but it to be more precise, you would say it's to the advantage of the virus to have this quality, because then it increases its chance of spreading, and therefore it can, maintain itself. But yeah, of course, it doesn't have true intentionality.
But even maintain itself, I mean, is it concerned about maintaining itself?
No, it has no concern. Nope. It's
Yeah.
it's a chemical entity. It's not a molecule. It's a collection of molecules with surface molecules that match certain receptors and enzymes that work in our cells and work in bat cells, and it's just it's a package that that has all the components needed to replicate itself. And it's like life will find a way is one of the things you hear from film. It's life has a way of achieving its ends, especially when sort of survival is on the line. So there's a very strict selection for replicating and surviving. And so viruses that mutate in such a way that they can't live, they just don't. They're gone. You have to, and optimization and can occur, things like this. This is just what we see in nature, this process. And it's easy to say, oh, virus wants to do this, or my cat is joining me here. Come on, get off of there. And but yeah, it's certainly somehow it's easier to use that language, but it's definitely not precise scientific language.
Mm-hmm.
Craig, as the virus RNA, this particular virus RNA is replicating itself throughout all of the RNA that it has, would it be accurate to say that each generation there may be a mutation somewhere along the replicating line, and it would be eventually happenstance that the modification of the RNA would hit the particular section that is manufacturing the protein that is doing the damage in the host organism. So it's really happening all the time, but it's only a chance that that a natural mutation might occur. And could that ever be, that particular section of RNA be identified as to where it is located? And could it be, could the disease then be attacked from that particular point of altering the actual RNA with radiation or something?
Right. Yes. So, so let me show you a picture of the virus. I'm going to share a screen, and you'll see all you guys will shuffle off to the side, and you'll see my other screen appear on your computer. You guys see that?
Yep.
Right. All right.
Yep.
So this shows. Let me make it a little bigger here. Can I do that? Yeah, a little bigger. So this shows a cell. This big block is a cell, and here's the coronavirus attaching through the spike protein, and it's binding to a receptor on epithelial cells. And then the genome is a RNA genome. But then there's an RNA-dependent RNA polymerase, and it makes RNA from RNA template. It encodes its own RNA-dependent RNA polymerase. And this is one of the genes used in some of the test kits. They detect this gene right here. And then several transcripts or RNA products are made that are then ribosomes translate that into proteins, and the proteins.
Does that go through an arrangement of the amino acids then?
Yeah. So the amino acids are the recipe for which the order of amino acids is encoded in these strips here. These messenger RNAs, right? See, mRNA synthesis, and the whole genome itself also is replicating, and then the eventual. So I guess this is the genome right here. That's going to be packaged. See it down here?
Yeah.
Get packaged, and these proteins package it together into these new virus particles, which then bud out and release. And so it's not like one gene is killing the cell or a bad thing. It's like this whole process kills the cell. Typically, they bust out of the cell, potentially. I'm not 100% sure of that with this virus, but so viruses are a very big, diverse group, and they all do slightly different things. But this is an example of SARS-CoV-2, which is the actual COVID-19 virus, the actual name of it. So yeah, I thought you'd find that interesting. Now, in terms of the genome, it's pretty big for a virus. It's around 30,000 base pairs of RNA. And you can see the arrangement of the different genes, and they know what some of them do. But yeah, so lots of information out there. You can compare viruses. One of the things I wanted to mention is that
did I hear you say 30,000?
Yeah, 30,000 base pairs. Yeah, so it's a big genome. Yeah, some of the viruses like chickenpox virus and some other herpes virus family members are also really big like this, and they have hundreds of genes. This is not as big as that, but again, the point is that viruses are a really big, complex group of very diverse things. Won't call them organisms, but they're not considered organisms. They're considered viruses. They're less than a cell, but they're more than a single chemical or more than a single protein. They are distinct evolutionary entities that you can map onto family trees. Where do I have here? I got to go back to the screen share. Let me show you this.
Is there a name for that status or that phase between inorganic and organic?
Well, it's a virus. It's definitely organic. It's lifelike. I think the debate over whether it's alive or not isn't really all that interesting, because we know what it is. We know how it works. It's really just whether you choose to call it alive or not. Okay. See what I mean? And so here's these little branches are different clusters of sequences of sequenced viruses, and you can see that some of them are known to cause cold viruses. In fact, the very first coronavirus was discovered by a woman in England. I heard this on the radio yesterday, and she was working in collaboration with someone who was studying human common cold, right? So common cold is caused by a number of these coronaviruses. This particular one is, all of these are the COVID-19. They all are very, very similar. See how short there's no horizontal bars because they're basically identical. And then this one right here, this little bar here is SARS, right? And then this collection here, down here, are all from bats, right? So the SARS looks like it is very closely related to this one that's known to be a bat. And all of these are bats, right? And see this branch of all the coronaviruses? They're close to bats. So we think that the coronavirus came from bats, the current coronavirus, I should say. But people have no idea how much, the average person on the street has no idea how much is known about viruses. And we've probably just scratched the surface of what's out there. Although with genomic technology, we can actually sample what's in the environment, and it's to our advantage to do so. And who knows, maybe even collect every bat coronavirus known and make vaccines to them in advance based on some kind of formula that we know works for SARS or these others, It'd be interesting.
So the destructiveness of this particular virus then is just the mechanism in which it goes through in replicating itself then primarily.
I'd say that's generally true of viruses in general. Yeah, that's a general case, that they wreak havoc with cells and for example, the simplest virus is, well, no virus is simple, no cell is simple. One of the first, most well-studied viruses were the bacterial viruses, bacteriophage they're called. And when you infect bacteria with phage, it kills all the bacteria. And then you have a ton of virus, a huge amount of virus, 10 to the 10th copies per mil or something like that. Huge numbers. So, all right.
Do you want to talk about the
Just
receptor site?
Can I ask a question here? Having just seen that whole list of viruses, when they talk about being able to trace where one of those has come from, like from Europe, what are they looking at there? That what lets them know, or that this one must have been around now?
Yeah,
Something
that's
that was here already in December. What are they looking at?
They know that the one from that came to the U.S. came from Europe, not directly from China, and the reason is because, excuse me, there are probably literally 3 or 4 point mutations, individual DNA, well, it's RNA, individual mutations in their genome. And you can just say, oh, look, all the ones in the United States have this, this, and this mutation, which was seen in Italy, but not seen in Wuhan.
Okay.
That's how they do it. And for example, I know a friend of mine who's in Washington State was following this, and they knew that there was community spread happening by looking at the genome sequences of several virus isolates that they found early on. I think there were 3 or 4, and you could see that like they all had this one set of mutations, but then there was half of them had this other one, which means based on the known mutation rate, positive mutation rate, that there was enough of it spreading around for this period of time long enough to accumulate additional mutations. So the fact that the early isolates were not identical meant that it had been circulating for a period of time.
And the slide you just had up, which showed where all these, like the bat viruses and all these other, were those all changes via mutations?
Yes, yes. So I did want to mention that someone brought that up earlier. This particular virus doesn't mutate as quickly as lots of other viruses. And the person who sent me those slides had mentioned that they think there is a actual proofreading polymerase that, in other words, one that's quite accurate. Some viruses on purpose make lots of mistakes so that they can evolve more quickly, but this one doesn't seem to have that strategy. So that's good for vaccine development, because you don't want to have the virus changing itself very quickly, because then it could escape, it could develop mutations to avoid the vaccine and that kind of thing. There was one, I wanted to show this. This is the current status of, let's see, January. No, that's too long ago. So this is an animated graphic. You can see January 21st, January, that's really early days. And you can see the confirmed cases. These are confirmed deaths. Whoops. And if you trace this, and I'm sliding it along, you can see different countries. See how China leveled out right up here. Whoops, China leveled out. United States. And this is January 21st. Let's keep going here. Let's see. Bring it up to, does it only go up to May 7th? Well, that's, wait, that's today. Okay. Yeah. So here we are. Here's the United States. All right. So we are leveling out. And Italy is leveling out. A lot of countries are leveling out. But the thing is, we have not achieved, we don't think we have achieved a high enough level of Japan. That was an interesting one, where it sort of leveled out and then jumped up again.
Craig.
Yes.
I saw someone compare one graph that just focuses on New York. And if you take New York out, and if you take New York out, you see the graph for the rest of the United States going, not plateauing, but going up. Have you seen that?
Right, because they're doing a pretty good job in New York. I haven't seen that exact thing, but it makes sense. So it's very different in different states. One of the best things I saw was a New York Times video. I saw this 2 days ago, I think, or yesterday. I'm not sure when. They talked about, I believe it was Ohio. Ohio has a very, very wonderful like public health
Yeah.
official, a woman doctor, who I wish I could remember her name. And she has achieved mythic status, hero status, because they've had some like 600 deaths next door in Michigan, I think it is, which has less of a population, has like, I don't know, 12,000 deaths or something. Where is it? New York, Mass, California, Louisiana, Ohio. Okay, so Ohio has 1,272. Where's Michigan? Michigan. Is it not here? Yeah, Michigan. 15,000 recovered.
Wow.
4,000 deaths. Ohio, 1,272 deaths. It doesn't say recovered. I wonder why it doesn't say recovered. The number recovered is just blank. There's a drastic difference, and it's because they were shutting down and leveling the curve weeks and weeks and weeks earlier. And so that's the interesting thing is the dynamics of how early action plays such a big role, which is why it's concerning that people are ready to go back to normal.
Yeah.
It's premature, because we're going to see the curve start to go up again.
Yeah.
Any other questions? Did I answer your questions, you guys? It's fun.
Craig, in terms of vaccine development, that we have already developed a vaccine, we're testing it now. Is that some kind of a record?
Oh, yeah.
Development of a vaccine. I mean, how about HIV and the fact that we've never developed?
Yeah, HIV is a very unique case. Retroviruses in general are much, much harder to make vaccines against. One of the reasons of many is that it mutates really, really quickly. So it has very, very high variability in the population. It also has a tiny number of genes, I think about 10 genes, and a much smaller genome. And also, most vaccines work by sort of mimicking the disease in a way, sort of, whereas if you were to, like, you could never use it's hard to imagine using a live vaccine, because this particular virus infects the immune cells, right, directly. So there's a number of reasons why HIV is particularly challenging to make a vaccine for. On the other hand, with colds, we think they promote fairly long-lived immunity, but that's really not fully known. Anthony Fauci says, we gotta watch. We're not sure. We don't have the data. We don't know for sure if this vaccine is going to be long-lasting. It might require a booster shot. It might require 2 different formulations to work effectively, but I'm pretty confident that even if it's not a perfect vaccine, it will make a great improvement. It's not like dengue. So people say, oh, some vaccines make the disease worse, and they're thinking of dengue. And dengue is a special case where you have 4 different varieties of dengue that are similar. And if you have 1 or 2 of them and then get the 3rd, you have much worse disease. So that's a special case, which we don't think the coronavirus has that kind of a situation.
Okay, just something else. I'm wondering, are you aware of the tweet, of Obama's tweet a couple of weeks ago, in which he said, we continue to wait for a coherent federal plan. But then he said, he cited Massachusetts in a New Yorker article as a state that has a 5-point plan that is what he was holding up as the plan that the federal government should have and every state should have. And I don't know whether you have the testing capacity, but what is actually happening in Massachusetts?
Yeah, I'm in Massachusetts, and every single day, Governor Baker is giving an address. I think that's happening in a lot of states. And he's taking it quite seriously. And we've just had, for example, this week was mandatory mask wearing whenever you're unable to, distance. Just like you don't have to wear it in your backyard, but if you go out, you have to wear a mask by law. I think it's law. I think local authorities are given the ability to put a fine on you or something like that. So we're taking it pretty seriously, and we didn't have as high the surge as people had feared, but the cases are still high. And on a daily basis, let me tell you, people in the hospitals are, very, very busy. And I hear, the obituaries, so-and-so died, this and that. It's happening. So anyone that still holds to the idea that this is just like the flu, the numbers don't lie, and the obituary pages don't lie. So this is a very serious illness. It's novel. We have no immunity to it, and until more people have immunity, whether it be from getting it and recovering or a vaccine, hopefully a vaccine, then we really have to be very careful. Now, it might be the case that, with everyone wearing a mask, maybe that will really diminish the spread. It seems to. It's hard to know how much of the curve flattening, if you will, is due to just staying at home and how much of it is due to wearing a mask. Again, it's those little spittle droplets. It holds them in. It holds those big, relatively large droplets from flying out. That's a big plus. And I was out today. I'm planning to move a beehive to a neighbor down the road that I just met today, and she's an older woman, and we were going to go look at another site, and she's like, well, can I just jump in your truck? And I was like, no, I don't think that's a good idea, because I went shopping earlier this week, and maybe I caught the virus, and these little droplets can fly around in a vehicle, so it's better if you take your car and I'll take my car. But people don't really have a concept of how it spreads. I wouldn't want her to catch it from me, so I kept my distance and wore my mask. So. Excellent.
A rather germane question, but related to what you were just saying. So if one goes to a grocery store, is it incumbent on when you come home to put all your laundry in a machine and wash it?
I don't think so. I think that might be overreaction. If there's a real super spreader in there, maybe. But I think that there's always risk to everything we do. You drive in the car. It's a matter of how much risk is reasonable and how much is easily preventable. I think if you work in a hospital around people that are infected, yeah, you should do that with your clothes. But presumably most people in a shopping situation are not infected and spreading. So I would definitely avoid, touching my face. I would use hand sanitizer after getting out of the store. I would maybe set my mask aside, launder that, that kind of thing. But yeah, I think that's probably it's not as easy to catch it off probably a surface like a clothing surface, because it's so three-dimensional. And yeah.
Okay.
Greg, a question about when the first several weeks went by, they said, this is a disease of the lungs. And then they said, oh, this is a disease of the lungs and the kidneys. Why did it pick those two?
I hadn't heard the kidneys. Honestly, I hadn't heard that.
They're both the sites of manufacture of angiotensin converting enzyme, which is the receptor for the virus.
Okay, well, I heard that the intestine can also, like epithelial surfaces in general. Yeah, the other thing, they're learning a lot of new tricks, if you will, the things, features. So one of them is blood clots and strokes. It's not a pretty picture. Have you heard that one? Have you heard that one about the strokes?
Yes.
Yep.
Even young people, right?
Yeah, exactly.
And this new pediatric thing.
Yeah, there's the pediatric thing where what was it?
It looks like
Kawasaki
a
disease.
multi-system disease, which includes a rash and cardiopathy and sometimes fatal disease.
Yep, and explaining that is challenging. Whether it's the antibodies being produced that are reacting and depositing in the tissues, and if maybe the antibodies are produced are causing the virus to be dead but clumped together in little clumps. Those clumps, if they stick in the tissue, can activate complement, which is another aspect of the immune system. It can cause all kinds of local inflammation, so it could be something like that. But it's not everyone that gets it. There's also the foot rash, which looks to me like antibodies that are precipitating in the colder tissues, like the skin or the toes or the fingers and toes. You get the, what do they call that? There's a name for that thing that you get, frostbite type thing. Terrible. But yeah, it's definitely weird virus.
So Craig, if you were to itemize from most important to less important cautions for all of us, say 3 to 5 cautions, how would you prioritize them?
Stay in bed all day.
I think you want to stick with your close circle, and really minimize contact with people that could have been exposed. In theory, you're all sort of isolated in this community, and so you probably are fairly likely to not be carrying it because it's been over 2 weeks that you've been there. So, is it safe to mingle with each other in person if none of you have gone out and or had a possible exposure? It probably is. So, but on the other hand, there are people that are saying, well, like, I was really careful and I still got it. So, are you guys going shopping, or do they? That's one of the ways I go out to go shopping, the designated shopper. And you're going out and go to the post office and there's another opportunity, so I think all of us could potentially catch it. So anything you do like that, you just have to try to be conscious of what you're touching and then decontaminate those surfaces. So that's what I do.
We have our groceries delivered.
Right.
They're strongly encouraging us not to leave campus, but some people have had to visit their doctors, have had to even be in the hospital.
Yeah.
We had someone who was going to donate blood, and so.
Yeah, so all those are reasons why you wouldn't want to hang out with them for a couple weeks.
Yeah.
But that's good that they're doing that. I mean, it's scary, but you can't be paralyzed with fear. You have to live your life, and just try to minimize the chances where you can.
Are you still roasting your mail?
I am. I'm roasting it. It's such a habit now. I cook my mail. Who knows if it works? I think it probably does. It's quite toasty in there, and this is a virus that probably doesn't like to be heated up to a certain temperature, 150 Fahrenheit, somewhere in there.
So what is it that you do?
You put
I toast my mail in the oven.
Oh.
to what?
15 minutes.
150?
Yeah, I put it on a little above the warm setting. Just I think that it's not enough to turn everything brown and get it to catch on fire, but it's enough to kill off the virus if there is any. And I know the postman has gloves on, but he's got his gloves on, then he's like, and then he's picking up, you just it's nasty. This stuff goes everywhere. So I'm just taking every precaution. It's not any really extra work to do that per se.
We have all of our groceries delivered to the door, but even so, somebody, the stock boy, put the cans and the
Yeah, I know.
packages on the shelves.
Yeah.
The shopper went and took them off the shelves and put them in the basket. Then she goes to the checkout where the lady touches everyone and rings it up. And then she brings it to our place at the checkpoint where one of our staff, who also lives in the community, picks up the bags and carries them to our door. So there's just no way to have, we don't have any direct contact, but there's secondary and tertiary and primary contact. It's just unavoidable. So we try to spray and wipe everything down as much as possible.
Yeah,
We haven't gotten to the point where we wipe every grape, but the package they came in.
Yeah, you can you rinse off your grapes, and you know,
Yeah.
I think the other thing that will be as good is if someone in that chain comes down with it, you'll hear about it. You know, they'll tell you, and then you can take greater precautions. But it's likely that they're not sick, but there always is the chance. Yeah, there's always a chance. Places where you're most likely to spread would be in choirs and choruses where all these little spittle particles are going everywhere. So, you know, just transferring groceries, maybe not the most efficient spreading mechanism. So I think Fauci would say not to worry too much about that. But yeah, have a reasonable level of concern.
You have to eat.
Yeah, you do. Yeah.
Can't give it up.
Yeah. And usually those people are wearing gloves, and they're spraying their gloves, and they're doing everything they can to minimize the spread. Well, I have to go pretty soon. Any final comments or thoughts?
Well, certainly a thank you.
Yes.
Thank you very much.
Craig, that first visual you put on was
Hello to Kelly.
exceptionally well done and very clarifying, at least for me. So thank you for that.
Oh, you're welcome, Dave.
Why don't we look at two weeks ahead of now? Because things are happening so fast, there might be something worthwhile looking at. Does that sound?
Yeah, sure. I mean, 2 weeks or a month. It'll be interesting to see because maybe a week ago or maybe this week, a bunch of states are opening. I mean,
Scary.
but a lot of people are wise to that. You know, they're taking precautions. So they may not see a dramatic uptick, but they'll definitely see some. I would suggest. So yeah, it's very spreadable. All right, you guys. Thank you. You're welcome.
Enjoy your spring.
Thank you.
Enjoy meeting you. Maybe someday in person. I'll come down there to PA and see you guys.
Be
All
great.
right.
Bye-bye.
God bless.
Bye all.
Thanks, Craig.
Bye.
Put your comments in, Lola.